So hi, I'm Alex Hoischen. I'm a professor for Genomic Technologies in immune-mediated and infectious diseases. And I work at the Radboud University Medical Center, which is in Nijmegen, the Netherlands. While we get better and better at the genetics of rare diseases, the inconvenient truth is also that we don't find the genetic cause in all our patients. And we even have cohorts where we're really convinced there must be a monogenic defect, and still we don't find the mutation. And we think that in part that is due to technical issues with the current tests. So we think that long reads may be more accurate and may be able to help us find so far hidden mutations in the genome of our patients. For example, we worked on the genetic odyssey of a family that witnessed full tragedy. They lost two of their daughters due to a terrible early onset brain tumor. So there was a strong suspicion for a genetic disease. We even knew the gene, the locus, but the standard tests that were performed at that time missed to find the mutation. And then we applied long-read technologies, in this case the combination of HiFi sequencing and optical genome mapping, and we identified previously missed and hidden mutation. So we were able to finally diagnose that family, truly on a genetic diagnosis. And even better, and that brings me to the memorable part of that case, my colleagues in diagnostics were able to develop a custom-made prenatal assay for that family, and that allowed us to offer the family a prenatal test. And then the mother got pregnant again, and the test was luckily negative. So that family now has a healthy baby boy, thanks to our technical innovations that enabled the diagnosis in first instance. So yeah, we by now have several cases where HiFi genomes now give us insights or find mutations that we initially missed. And I think we find a group of cases in which those are so-called structural variants, so larger chunks of DNA that are deleted or gained or placed elsewhere. And we also recently discovered several repeat expansion disorders that were initially missed. So that is very exciting and really starts to show the power of the long-read HiFi genomes. So in fact, we've now done a series of tests and studies. And very briefly, we did initially a retrospective study. So just asking the simple question, can HiFi genomes find even the most difficult mutations that a diagnostic lab has to detect? And that was extremely successful. In a second tier study, we looked at so far unsolved, undiagnosed rare disease cases. And in 10 to 15%, we found extra yield mutations that were initially missed. But then the real landmark study is just recently finished. And those were 1,000 genomes by HiFi genome sequencing that we performed in a blinded, prospectively designed study to mimic standard of care diagnostic testing for rare diseases. And also that result of that study was overwhelmingly positive. So by now we believe that HiFi genomes could become not only a first-tier test, but it harbors potential to become the generic, the only test we may do for the vast majority of rare diseases. So the extra yield we find from HiFi depends, of course, heavily on the selection criteria of your cohorts. So we have specific cases where great clinicians that we work with know very well the one disease locus. And in those cases, we almost always find the missing mutation. So there's some diseases where only one gene is really published and known to cause disease. If the standard assays didn't find it, now HiFi really finds it. And usually those are structure variants. We have several examples, tens of those where it's a so-called mobile element insertion. So those are individually rare and almost exotic events, but collectively in these unsolved cases, they may be quite overrepresented. And in fact, they are the reason why the standard assays didn't find it. If you then look at larger cohorts that are more representative for the entirety of rare disease, I think there we start seeing an extra yield of 10 to 15%. And that doesn't sound a lot, but every single percent of extra cases where you can provide a diagnosis is extremely meaningful and extremely important. So we now start seeing that HiFi genomes are more complete. Quite often, they're more complete than a variety and add-on of different tests that we perform standard so far. So I think it's fair to say, and I see that in my own research group, the postdocs that now started to work on HiFi genomes don't want to go back. So that already speaks for itself. I think that tells us they are fairly complete genomes. And it's in particular this fact that we can start phasing. We look at entire alleles. We are not looking at variants in isolation. And now something new, we also see methylation in cis to a genetic variant. So I think that's becoming incredibly powerful, and it's really nice to see that all in one assay. So as some may know, we were quite early on believers in PacBio sequencing. So we worked on Sequel 1 and Sequel 2. And with Sequel 2e, of course, HiFi genomes were already feasible and possible. So we started to enjoy the quality of those genomes. But the biggest limitation was price and throughput. And of course, that has really changed thanks to the Revio instrument. So we're now very lucky in Nijmegen to have already 4 Revio instruments. And by just using 3 of them, within the last half year, we cranked through 1,000 genomes. So this really becomes scalable and really allows us to do more meaningful studies to provide clinical utility of HiFi genomes. So by now having instruments that are higher throughput, like the Revio, of course, we also move our challenges, for example, to library prep. So by now, it is not feasible anymore to prepare libraries in a manual fashion. And I'm pleased to say that the last 1,000 genomes we processed in-house were fully automated library. So our very skilled colleagues in our diagnostic facility who really master automation, and now we have an extremely well-automated protocol. And honestly, 2 years ago, I would have not believed that the quality of automated libraries are so good. And we've really reached that stage that we now have fully automated library prep that directly goes on the HiFi sequencing, and they're really high quality. So that is quite incredible. And in fact, I dare to say this is not the bottleneck anymore. So of course, we have to realize that our lab in Nijmegen is huge, and we usually go for bigger and bigger machines, but not everybody can follow that trend. So I think with the Vega system, you now have a smaller instrument, and the initial instrument investment seems to be also quite a bit lower. So I think indeed, many other labs may now follow and perform HiFi genome sequencing and targeted sequencing on that system. So I think it was a good move to come up with a second instrument that really makes HiFi sequencing more available for more labs. So of course, with the new SPRQ chemistry, we can now perform sequencing for much higher throughput again. So we aim to run 2 samples per SMRT cell. So that's literally doubling the number of genomes we can do on a single instrument for the same price. So we're very excited by that because, again, more samples means more interesting clinical questions and faster implementation of that test. So yes, that is a great move, and we're really enjoying that. One thing to add is we compared what coverage we need to find very tricky mutations, and indeed we can see that 15 or 20-fold coverage HiFi genome is already better than many other tests that we've used in the past. So I think the standard of using 30 or 40-fold coverage in the field comes really from the short deep days where we looked at variants in isolation. So you really wanted to be sure that every variant is so well covered. With HiFi genomes, we can really literally see both alleles. We now really have the experience that 20-fold coverage may well be enough to provide already a very high quality genome. And in fact, in other research projects, such as SolveRD, we even only performed HiFi sequencing with 10-fold coverage and already made an incredible amount of discoveries on all variant levels. So we found structure variants, point mutations, indels, and repeat expansions, all with 10-fold coverage. But that, of course, is a research study. So I envision that for real clinical use, we get very high quality when using 20-fold coverage. So I guess if I look a little bit what we want to use HiFi sequencing for in the future, in the near future, it's really the increase in throughput. So I think applying it to more and more rare diseases, even making it a first-tier generic assay, is of course what we really want. So if we can really scale that to thousands of samples on a yearly basis, we not only provide a better answer than the current tests, but also the layer of discovery will be incredible in the next year. So I'm really looking forward to find more structure variants that we initially missed. And I'm personally very thrilled by structure variants that don't hit genes directly, but rather the regulatory part of the human genome. So I think there will be an incredible layer of discoveries that is coming in the next years. And there we really need a lot of genomes to start understanding that, and that now is starting to be enabled. Something else I'm excited about is using HiFi sequencing also in my own research, going beyond standard DNA sequencing. So of course, the current HiFi genomes also provide DNA methylation, so that's already a nice intermediate layer. But for certain indications, in particular rare immune diseases where we can get affected cells from some of our patients, we'll start utilizing RNA sequencing or isoform sequencing on RNA much more in the future. We will really move from DNA only to multi-omics for some of the rare diseases to really understand the underlying biology, and I'm quite excited about that as well. So I think the vision there is very clear. The future of rare diseases must be accurate long reads as not only the first-year test, but truly a generic test. So for the first time, testing may become almost boring in a sense that we don't need to speculate about the test to do for disease A or B or C, but there may be a single generic assay that we run in our lab that gives a better and more complete answer in all our cases. So this should really become the standard for rare diseases. So to others that want to get started along with genome sequencing, I can only strongly encourage that. Get started on it. It's not difficult. Once you get used to it, you don't want to go back. Long reads are very exciting. And as said before, it's really helping our patients, and it's really helping you as a scientist to make new discoveries that were impossible before.